everyone, and welcome to today's webinar, Innovative Immune Monitoring, Bringing a New Dimension to Cytometry. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Talon Biomarkers. To learn more about our sponsor, please visit talonbiomarkers.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I would now like to welcome our speaker, Dr. Pratip Chattopadhyay, CEO and founder of Talon Biomarkers and CSO co-founder of TerraFlow Bioinformatics. For a complete biography on our speaker, please visit the presenter window at the left of your screen. Dr. Chattopadhyay, welcome. It's so nice to have you. You may now begin your presentation.
Thank you, Christy. It's great to be here. So I am pleased to talk to you about some of our recent work and what our companies do. And we're excited to sponsor this webinar with the folks at LabRoots. So again, the title is Innovative Immune Monitoring, Bringing a New Dimension to Cytometry. And so first I'll tell you a little bit about our company, Talon Biomarkers. We're really a head to tail, or a beak to talon CRO, beak to tail CRO. We start with study design. We can do the lab work also. We can do data analysis and interpretation. We're not just a set of skilled hands, we're actually your scientific partners also. And we've been in business for 4 years now. We've got a roster of delighted clients who really feel the power of the innovation that we're bringing and are happy to have us integrate with their translational science teams. So specifically in terms of our services, our primary service is high parameter flow cytometry. We also do single cell sequencing, and particularly with technologies like CITE-seq and AbSeq, which replace the fluorochromes on antibodies with oligonucleotides. So it's single cell cytometry and sequencing, molecular cytometry, if you will. We do cell sorting and capture for RNA sequencing, ELISpot ELISAs. We can process samples, and we have a robust effort in AI and algorithm-driven discovery and data analysis. We also have tools that we've developed for automated data monitoring in QC, and we help you with study design and implementation. And there's lots of consultation and support that's built into our work, that's built into our fees and our charges. We really believe in leading with the science and doing the science first. And we don't just dump data on our clients. We are eager to see the project through to its fruition. So we're not just a CRO, not just a contract research organization. We really see ourselves as a knowledge resource organization. We are not just a set of technical hands. We're your partners who are driven by quality, outcome, and particularly interpretability of the results. We have an important focus in that. We lead with innovation. We do high content work so that we maximize the information that's available from every single sample that we're getting. We're efficient in terms of our cost structure, and we're robust because of all the quality control that we do. There really isn't an experiment in modern flow cytometry that I haven't had a hand in helping to develop because of the hardware, reagents, and software that I grew up developing at the NIH and then later at NYU. And so we're really leaders in the field, and we're proud of it. And we look forward to talking with you more about your needs. We're also real connectors. We enjoy the idea of being keynotes in a network of curious folks and generous idea generators. So our network consists of top scientists and vendors who share our values, and we bring those folks to you as partners to help in doing the work. And we can engage the partners so that our entire collaborative effort is maximally rewarding. So our guiding principles are flexibility and adaptability. We don't require unnecessary steps or controls in projects. All our projects are custom quotes. No need to tailor your unique research to a particular box or kit. We have experience adapting to big pharma vendor and IT requirements, and we have a strong thread of social and community responsibility in the work that we do. Many of our computational tools have been built by interns from the Morris County Vocational Technical School and the Community College of Morris County. We have here in New Jersey, we have a New Jersey Economic Development Authority grant, and we're also heavily interactive with the local trade association, BioNJ. So today I'll talk to you about the work that we're doing where we're innovating at every turn, and we expect that a lot of folks who do flow cytometry are going to pick up on these ideas, and we're very happy for that. We're very happy to advance the field and help people with the flow cytometry they're doing. It's not just about turning tables and getting business as quickly as possible. It's really about helping people maximize the flow cytometry that they do. And so today I'll talk about panel design. So how do you choose the combination of antibodies that you can use in your study? I'll talk a little bit about data analysis and new methodologies for data analysis. I'll talk about interpretation of the data and a very unique platform that we have for that. And then I'll tell you about what's coming up in the future, a really exciting new technology that's come available in recent years. And we have a partnership with the folks at Lay's Innovation doing that kind of time-lapse flow cytometry that we'll talk to you at the end of this lecture about. So let's start by talking about panel design. And this is a really key element of how you do high parameter flow cytometry experiments. And there are different approaches for doing panel design, some of which I actually helped develop and sort of proselytize about in the early 2000s and in the 2010s. The first is to look at spillover, the overlap between dyes. And this is essentially looking at the compensation matrix that's generated to remove the overlap between fluorescent dyes. This is a really easy way of panel design. And the idea here is that if you have a lot of spillover, then that's two sets of reagents that aren't going to work well in your panel. But that's not necessarily true, because high compensation doesn't mean the resolution of your flow cytometry experiment will be poor. In fact, compensation is meant to remove that overlap. And in any high parameter flow cytometry experiment, you're going to have compensation. It's unavoidable. Also, compensation will differ by the dyes chosen. So this idea that we can just look at spillover is kind of a holdover from the late 90s, early 2000s, where there weren't that many dyes to choose from, and we weren't doing more complicated experiments. So it's really kind of a relic of the past. Nowadays, with spectral cytometry, people are developing metrics like similarity, and then telling you that the overlap between dyes can't be greater than 98%, otherwise it can't be unmixed. And these types of parameters are available in software and web tools that are easily accessible. But the DIY calculation, if you do it in your own lab with Excel or something like that, it's not easy to do. And it's not specific to your instrument. It's entirely dye-based, and it's just based really on pairs of dyes. So it works reasonably well, but it's not the best solution. The third solution is a complexity index, which is really not too much more different than the similarity index. It's also available in software and web tools. We use Thermo Fisher's Bigfoot as a sorter here, and that has a wonderful system for calculating complexity in panels. It's very hard to complicate. The math that underlies it is non-trivial, and it doesn't, again, consider the instrument performance. It sort of gives you a universal answer, which is kind of good in some ways as you move from instrument to instrument, but it doesn't necessarily guarantee that your results will work, your panel will work on your instrument. So a fourth system uses spreading error, and this is one that we introduced, that I helped introduce in the late 2000s. So we have a tool that's called Color Wheel that calculates spreading error and considers the density of antigen on the surface of cells. It's specific to your instrument's detector performance. The calculation of this is complicated, but we have a software for it. It's basically a system that, for all intents and purposes, is guaranteed to work for your individual cytometer. And so that's the power of this system, and that's why we used it for so long. Now, it turns out the modern flow cytometers that are coming out now are really great instruments across the board, across all of these vendors, and they have pretty low noise in their detectors. And because of that, the spreading error is at least slightly less of a problem than it used to be. And so it may be that doing spreading error is kind of overkill. So what we really needed here at Talent was a fast method to build panels that would be accessible to staff with minimal training. We didn't want to wade through gigantic matrices of this dye versus this dye, this dye versus this dye, and so on. We wanted to find the best set of dyes for our instrument and rank the dyes on our instrument for those times when the ideal antibody conjugate isn't commercially available. And so the key words here are that we need speed and efficiency in the tool that we developed. And so the kids at the Morris County Votech School helped us develop a tool called Dye Diver, which automatically discovers the singly stained compensation tubes that we collect from our regular experiments. So it's just using as a basis the data that we're collecting over the course of our work here in the cytometry lab. And then it transforms the data so that for each channel, lower negative fluorescence equals 1, while the higher signals all equal 100. And because of that simplified transformation, we can really see where the problems are in potential panels. It then automatically generates a report. There's an instrument-specific transform spectrum that you can view for every dye. We can rank the brightness of the dyes, and we can calculate what's what I call the wingspan of dyes to sort of harken back to our name, Talent biomarkers. So the way that Dye Diver works is actually really simple. You can see here on the right side of the screen, there are 4 dyes that are viewed in a kind of existing or classical spectral viewer. And these dyes are PE-CF594, PE-Dazzle594, PE-EF610, and PE-Bio615. They're all read basically on the same channel in many instruments. And when we look at their spectrum with a standard spectral viewer, they all just kind of overlap. And so we can't really assess much from a standard spectral viewer. When we do our transformation, our binary transformation, where the low values are zeroed out and the high values are all pinned at 100, we can see that these dyes really differ in how much they spill over into other channels. PE-CF594, which is a perfectly fine dye on its own, spills into lots of other channels. PE-Dazzle594 also has this problem. PE-EF610 is slightly better. And if you look, PE's Bio615 actually has many fewer peaks. That's all shown on the left side of the screen. And so what we can do is quickly examine and tell the differences between these synonymous dyes. We can actually reveal the difference between these dyes. And then if we calculate these zeros and 100s, we can get a sum of the transformed signal in each of the channels, which is the dye wingspan. So the wingspan of PE Bio615 is going to be low because it doesn't appear in that many channels. Many of the channels are zeroed out. PE-CF594 is going to have a significantly higher wingspan. And the math that's underlying this is very, very simple. And so that's how Dye Diver works. Let me go to the next slide. So Dye Diver in action, we use Dye Diver in a study of acute myeloid leukemia, which is a complex disease with poor survival. The leukemia cells after therapy are considered measurable residual disease, or MRD, which can be a prognostic indicator. With clinical information, MRD can stratify risk and serve as a clinical endpoint. Flow cytometry is one of the methods for MRD detection. The other is molecular characterization of the AML clones. A common approach when we do flow cytometry, rather than the molecular characterization, is to focus on multiple small panels that look at the early maturation of stem cells into leukocyte lineages, Also look at myeloid precursors and how they're turning into myeloid subsets, and then look for abnormal phenotypes, abnormal expression of T and NK cell antigens on progenitor cells. Our client had a goal where they wanted to inform go and no-go decisions on an AML drug quickly. They wanted to get the most possible information about the drug's activity without taxing their internal staff and resources. And in particular, they wanted to determine if CD123, an expression of their molecule of interest, which in the interest of confidentiality, I don't want to indicate here, could correlate with disease outcome. And so Talon's response was to adapt a 27-color panel that was reported in the literature for the client's work. And we optimized this panel quickly for CD123 and expression of this other molecule using DyeDiver. And then we developed a rapid data visualization tool to eliminate the painstaking workflow of analyzing AML data and to determine essentially the pharmacodynamics of this drug. And so what we're doing here is not using these multiple panels that are commonly used. We're using a single 27-color panel that was developed by Paul Wallace's team and reported in Cytometry Part A. The challenge, though, was that this 27-color panel was developed on an Aurora, and we were doing our work on a Thermo Fisher Bigfoot. And so since it was developed on a different instrument, could we make it work? Well, because of the way that we do our experiments and the care with which we take in titrating antibodies, etc., we could really easily translate the data from the Aurora to the Bigfoot. And this is data from the Bigfoot showing the different populations of CD45 positive, CD34 positive cells, and a slew of other populations that we can identify within the panel. You can see that they all separate really well, and that you can find these really clear clusters of positive, negative, and dim cells. That's exactly what you're looking for. But there is a problem with CD123 and this other molecule that they're interested in looking at. The spreading errors are pretty huge on our instrument for this particular combination. The spreading error, in fact, on CD123 goes up to 10 to the third, and the spreading error for the other molecule goes up to 10 to the 2. So I've drawn these red lines here to show where the spreading error is, even though there are, for this molecule, no co-expressed cells in the sample. There's no co-expression in the sample. Oh, sorry, let me go to the next slide. Okay. But the problem with CD123 and this other marker is that there are just too many dyes that are packed around the CD123 region. And so at CD123, they're using SB436, CD4 is using PAC orange, CD13 is using BV421, and CD41 is using PAC blue. And we can use our dye developer to look at these patterns, and we can see that they're all kind of jammed up together with lots of peaks. And so if we combine these wingspans, we can see how much signal is overlapping here that's contributing to the spreading error that's occurring in the CD123 channel. So we reconfigured the panel to keep CD123 on SB436. That reagent was working really well. We moved the CD4 PAC orange to BV615. We moved CD13 from BV421 to R718. And then CD41, we moved from PAC blue to BV711. And now the wingspan in the same region where we're measuring CD123 is very simple and has gone down. And when we do that, you can clearly see the cells that are positive for both CD123 and this other molecule. I've left the red lines to indicate the spreading error that was in the previous panel, and you wouldn't be able to see this population if you'd left the panel as it was. And so you can see that now we're suddenly able to clearly define this population, and we did that very simply with a simple metric. We can go on and basically calculate the wingspan score for each of the detectors that's the primary detector, for each of the dyes that's the primary dye on a particular detector. So on the x-axis here, we have all the detectors that we have in our instrument, and on the y-axis, we have the wingspan score, and then these particular lines are showing you the wingspan for each of the dyes that we have in stock that we've measured the wingspan for. And when you do this type of analysis, you see some really interesting things. For example, we don't often use PE-Cy55 in our panels, but we would like to use a marker in this region. And we don't often use PE-Cy55 because, first of all, there aren't a lot of reagents out there available for it. And secondly, look at this wingspan here. It appears essentially everywhere. And so it's got a very, very high wingspan score. In contrast, in the neighboring detector where no other dyes are measured, Novafluor yellow has a very, very low wingspan. And you can see that here, Novafluor yellow 690. And so we've essentially replaced PE-Cy55 in our panels with Novafluor yellow 690. And we can make decisions like that based on picking out these high wingspan dyes and finding another alternative that's available. And there's so many reagents that the different reagent manufacturers have developed that now we can do this kind of work to make our panel building easy. So a client need was solved, a new tool was invented. Our proprietary new tool is unique. It's automated based on data already collected on site, an evolving library that accounts for newly introduced dyes. It's instrument specific. It helps improve existing panels and build new ones, and it makes it easier to add markers quickly, provides a list of the best dyes. It's simple, and it's actually part of a larger suite of automated tools called CytoSor for quality control. We're picking out the areas of quality control where there's a lot of manual and expert labor involved, and we're automating those in new and unique ways. I'm not going to talk about that today, but that's a strong element of what we're doing here at Talon Biomarkers. So I talked a little bit about panel design. Now we'll move on to data analysis. And with this AML project, the client had an important need. They needed a rapid system to determine drug effects without the complexity of hand gating that's done nowadays in the AML sphere. AML hematopathologists and generally leukemia hematopathologists are amazing people to me. It's really a labor of love how they delve into the data so carefully and so deeply. And what they do is they have to set up lots of different gates that they overlay to assess what populations are present in a sample. We took a slightly different approach that was well, not slightly different, it's considerably different. We took a considerably different approach that was much, much faster, and I think will be much, much more robust. Despite the incredible expertise of these hematopathologists, we have an opportunity here to automate this type of analysis. So we've developed a new AML landscape viewer that we call PhenoFalcon. It shows the frequency of cells expressing markers that are in the leukemia-associated immunophenotypes classification scheme. And what we're showing here on the x-axis on the bottom of this graph here is all the markers that are in the leukemia-associated classification scheme. And then if it's gray, that means that that marker is either negative or dim. Like CD45 is usually dim. It's not negative in these types of samples. We gate out the CD45 negative events. And anything that's green is positive. And what you can see then is that you can calculate the frequency of every single possible combination of these different markers. And so that's what's here. You can't see it, it's too small to see, but these are different combinations of pluses and minuses, expression and non-expression for all of these different markers to build a phenotype. And I'm going to highlight a couple of interesting ones here. The data we've calculated out for pre-treatment, before treatment with this drug, and after the first cycle. And what you see is that CD34 positive cells that are negative for every other marker, negative or dim for every other marker, they don't change with the first cycle of treatment. They are not eliminated. These cells that are CD13 positive and CD117 positive are interesting because they emerge after the first cycle. And so they're not present after treatment, they're emerging after the first cycle. They could either be leukemia cells that have shifted in phenotype, or to be fair, because I didn't remove the normal expression combinations, they could just be normal cells emerging. And then over here on the far right, you can see that there are various populations, including one that's positive for many different markers, that are only found pre-treatment and are lost after treatment. And so those are leukemia clones that are lost after treatment. And so with this viewer, then, you can actually identify which populations are shifting over the course of the treatment, which populations are lost, and which populations appear. And so it's a much faster way of identifying the leukemia clones and doing this analysis, and allows you to more rigorously look at all the combinations of cells, cell expression of the leukemia-associated immunophenotypes. The second example from our data analysis work is particularly interesting and exciting to me. It addresses this issue of compositional data analysis. Flow cytometry data is inherently compositional. Each of the phenotypes that we measure is a proportion of all of the phenotypes in a sample. And because of that, there's a sum constraint, so that the total is always constrained to 100%. Without absolute counts, which often aren't available, you can have spurious correlations with outcome that can emerge, some that mask the true outcome or phenotypic correlations. Remember, in compositional data, because it's all percentages, as one population increases, another population has to go down reciprocally. And this is often going to happen in a really complex way. There's going to be one population that increases, and to compensate for that, a number of subpopulations may decrease, or the reverse can also be true. And it's hard to know which changes are actually biological or which are an artifact of some constraints. This problem was first described in 1897, but it's only now being addressed in genomics and microbiome research, and it really isn't used in flow cytometry. So what we do to address this problem is do what the folks in genomics and microbiome work do. We do centered log ratios that actually solve this issue. And so to demonstrate this, we took data from an existing 15-color data set from a pharmaceutical client. It was longitudinal data from a dose escalation trial of an immune targeting cancer drug. No blood cell populations correlated to dose or the tissue results in the client's limited analysis. We examined the phenotypes that were present in the data set in the CLR transform space. And we used a comprehensive approach on the untransformed data to compare it to the CLR transformed data. So we asked the questions: what is the minimum dose needed for the drug to have an effect on immune cells? It's essentially a pharmacodynamic question. We asked what the relevant time points for future analysis are. We asked what cell populations are impacted by treatment. And we asked, do the pharmacodynamics impact the response to therapy? And the results were really actually kind of stunning. Our CLR work showed that one particular cell type might be an important biomarker for the drug. It increases it's not present at the 1 mg per kg dose. And at the 3 mg per kg dose, it's kind of flat in all of the patients. But around the 6 mg per kg dose, you suddenly see that there's a longitudinal increase over the course of treatment. So as you get more of this drug, you see more of this population builds up. And the population, as I said, is undetectable at low dosages. And then when you look at 10 milligrams per kilogram, more of the responders, you see them down here in the lower set of graphs, more of the responders have this population than the non-responders. So it actually might be a biomarker of response as well, not just a pharmacodynamic, a population that's a sentinel for the pharmacodynamics. Interestingly, this subset shares markers with a population that is uniquely observed in the tissue of responders at the highest dosages. So this idea that blood is not a good surrogate for what happens in tissue is not true here. We can make blood measurements, and that's really important for biomarker analysis. And this population wasn't identified by the classical non-CLR analysis. So it's a real breakthrough for our client, and we're really proud to have innovated and presented a new way of approaching this data that gave us these kinds of important results that can be readily translated, because they're essentially looking at biomarkers in the peripheral blood. Next, I'll talk about interpretation of the data. And to talk about interpretation of the data that we all generate, I'm going to tell you about a platform that is from a company that I co-founded with Dan Freeman, and Ariel Ginsberg is also involved as the CEO of this company and helped us build this platform out. The company is called TerraFlow, and that's the name of the platform. And our tagline is kind of never wonder what you're missing in your data again. So flow cytometry has really exploded over the last 20 years. 20 and 30 parameter cytometry experiments provide intricate and beautiful detail about cells, like henna tattoos that are common in South Asian culture. But that beautiful data is really ruined oftentimes by poor analysis. Data analysis platforms can be a poor fit for our data sets. They're like putting lab gloves on our feet, which is not nearly as beautiful. Common methods are ambiguous and incomplete. You will often see people displaying data using TSNEs or UMAPs, which are beautiful displays of data and have great value. But the problem is that you never really know if you're capturing all the possible cell types that are present in your sample, because you have to tune these algorithms carefully, and because there's often a limited number of clusters that appear. The second thing is that there's no statistics that underlies this. You're really examining a group of samples versus another group of samples and looking for differences visually, at least at first, and that makes these tools subjective. And so there are some other issues with UMAPs and TSNEs that we won't go over right now, but suffice it to say that there are likely better approaches, and we've developed one of those better approaches. So what we do is we take the entire data set and we look at 5 markers at a time, and we build out all the possible combinations of positive and negative expression of those 5 markers. And then we get another set of 5 and another set of 5 and another set of 5, and we sample the entire data space that way. And when we're building out combinations of all the markers with all the markers with sets of 5, we then also do that with sets of 4, sets of 3, sets of 2, and even the single markers themselves. And then we relate these similar phenotypes essentially in a network, where each of the nodes, each of these circles is one of the phenotypes, and it's connected by a line or an edge to another phenotype that just differs by one marker. And so you have these neighborhoods of related phenotypes. And then the next thing that we do is we exclude very rare populations that are present at, say, less than 50 events across the entire data set. And so, so not just 50 events per sample, but across the entire data set, which means these are exceedingly rare populations. And they're not even the rare populations that you would look for in MRD and MRD analysis or an antigen-specific analysis. They're way more rare than that. The next thing we do is measure the association of the frequency of each of these cell populations with some outcome. Is it related to response to therapy? Is it higher in the non-responders? Is it lower in the responders, etc.? So we overlay that disease association on top of this network. And then you see that some of the cell types, some of the nodes have a strong relationship to disease, where other nodes have less of a significant relationship to disease. And there's different levels of this relationship. You know, in a neighborhood, you can hit on one particular population, in this case, the CD4 positive, GITR positive, PD1 positive, that has the strongest relationship to outcome. And then cell types that are adding or subtracting one marker have less of a relationship because there's a little bit of noise in that measurement then. And then we find the optimal phenotypes. We find the phenotypes with the strongest relationship to outcome. Those are representative of the neighborhoods. And then we basically report those in interesting ways. And we do this automatically. So you just upload the data onto a website, essentially. And in 24 hours, you get a complete preprint on your data back. And you get all this information that identifies the particular phenotypes, the explicit phenotypes. It's not just a couple of pixels on a graph that are red versus a couple of ones that are blue in another graph. You actually pick out explicit phenotypes that you can quantify and that the system quantifies and provides you statistics for. in this case for training and a validation set in this study. We also build machine learning models with all of the markers that are in the phenotypes that are interesting. And then what we do is we calculate the AUC, the sensitivity and the specificity of the full set of markers that is correlated with outcome. And then we go through and we drop each of the markers one by one and see how that AUC changes. And what happens is that you hit a set of markers where when you drop those markers out individually, the AUC tanks. And what that means is that those markers, and just those markers, are the key critical markers to discriminate the two patient groups. And so when we do that and we pick out a set of smaller markers, we can then determine the sensitivity and specificity of that new smaller panel. And this is all done automatically, and it's done on training and test sets with cross-fold validation with internally within the data that you provide. And so we really have a biomarker discovery engine here where we upload the data and then we find the major cell populations that are important. And then we have this exciting new feature where we link the cell populations to the literature to help us contextualize what those cells exactly are. So we go from phenotype to interpretation. And in this graphic, what we're showing is an example of that interpretation. I won't go, in the interest of time, I won't go into it with too much depth, but you can actually select the populations that differ across your patient groups and then basically hit the little down arrow button and you'll see the papers that report that same population in the same setting or different settings, and you can glean, it's highlighted automatically where those where a description of that population is presented in the paper. And so you can build hypotheses down the road. You can understand mechanisms of action by basically doing your interpretation from the literature. We are removing the requirement that you sit on PubMed and you scroll and you pick out things, and you think and you read. Thinking and reading is still really important. You still have to do that here, but we get you to that point much, much faster. And so, so here is an example with cell population that's FOXP3 negative, IL-10 positive, that was different in the study of COVID that we analyzed. And it's an interesting population because IL-10 positive cells are suppressive, FOXP3 positive cells are regulatory. So how do they overlap, and what does it mean when only a phenotype only has IL-10 positive cells, but not FOXP3 positive cells? Well, those cells are likely to be TFH cells with regulatory functions, as was recently reported, and the references are there. And so TerraFlow actually finds the papers for you and is able to, TerraFlow is actually able to find the papers for you and then identify the explanation as well to help you with interpretation. And I don't think you saw, but this is the population, FOXP3 negative, IL-10 positive, and this differs across the different patient groups, and that's the reported information. So, so we've covered interpretation in a really exciting way, I think, and now I want to transition to talking about the future of flow cytometry. Currently, we make a lot of static measurements in flow cytometry, essentially snapshot measurements, but the future holds more dynamic measurements for us, thanks to technology that was developed by Lay's Innovations, who is advancing biology through connected single-cell data. And we're working with them as a partner company because of our ethos of driving innovation in flow cytometry and biomarker discovery. All right, so let's imagine that there was a movie which I happened to find that's called Gandhi versus Jason, which is a crazy, just on the surface is a crazy movie that's out there. I've never seen it. Imagine that this movie was just a snapshot photo, though. What would you be missing? I mean, what initiated the conflict between Gandhi and the killer Jason? How and when does Gandhi fight such an accomplished killer like Jason? Is either of them terminally exhausted, or are they just catching their breath for the next battle? And in fact, we can think about this when we do our analysis of T cells versus tumors. Why are we only taking snapshots of an immune response? It's like a movie, but we're only looking at snapshots. What are we missing? What initiated the response? How and when does a T cell attack a tumor? Is an anergic cell really anergic? Is it really terminally exhausted, or does it just have slower kinetics for its response? So the next frontier in cytometry will connect the dots between these various single cell omics measurements that we make to provide maximal information about cell genotype, phenotype, and function. It'll flesh out the full story behind when a T cell attacks a tumor, or the full story behind an antigen-specific response, or whatever interesting disease mechanism is happening. It'll make disease mechanisms and biomarker discovery more efficient and fruitful. It'll tell the story of immune responses with dynamic measurements instead of today's static snapshots, and we can likely predict phenotype from function, or vice versa, to find the simplest biomarkers. The current tools that we have available don't generate this kind of connected and dynamic data set that's needed for training, for example, predictive models that can predict function from phenotype, or so on. So as we're starting to do more and more AI and machine learning work, we're going to need the data sets that help us to develop these in vitro modeling efforts, or in silico modeling efforts. And so the data sets now that we get from transcriptomics, phenotyping, metabolism, morphology, function, they're really distinct and they're siloed, and that slows our discovery and therapeutic development programs while increasing downstream costs, because you have to do all of these on different samples. If there's a way to track millions of single cells from genotype to phenotype over time to their function, we could truly, completely, and comprehensively analyze immune responses and learn the rules that predict phenotype or function, making biomarker discovery more efficient. And I'm really excited to work with the folks at Lay's Innovation who've come up with this concept and the conceptual underpinnings that I'm showing you of this, not just to mention this amazing new technology. And the new technology really involves optical cell barcoding. So these are like QR codes for cells that enable non-destructive, repeated tracking of millions of cells at a time. DNA barcodes are available, but they get destroyed. And so you can't repeatedly track a cell that's been barcoded with a DNA barcode. This can work with any cell analysis platform, flow cytometry, sorting, imaging, sequencing, where tracking cells would be needed. And what you get from it is dynamic single-cell data from live cells across multiple platforms. So these are the ideal kinds of data sets to train artificial intelligence or machine learning predictive models. So how does it work? Well, the optical barcodes are essentially what Lay's calls laser particles. These are micron-sized semiconductor lasers. What does that mean? Well, that means that they emit very sharp light within a very narrow wavelength. It's 100 times more narrow than quantum dots. And you can see the wavelengths of emission of 3 different laser particles here, and their emission spectra are extremely pure, unbelievably pure. The light is emitted in the infrared spectrum with no interference of conventional fluorescence. So what you need to be able to measure this is essentially an infrared spectrometer that's attached to a flow cytometer, and that's really what Lay's technology is, along with these laser particle discs, essentially. And so so these are covered in more detail in Nature Photonics and Nature Biomedical Engineering. But the important piece here for flow cytometry work is that there's a biocompatible coating on them, so you can stain cells with them. And there's antibody functionalization that allows you to tag the target cells. And let me go to the next slide the right way. So our vision is to generate high-value connected single-cell data sets, leading to rapid advances in developing predictive models for cell biology. You can imagine that we can unlock the single-cell potential across platforms using very similar workflows that just involve labeling these cells. We can connect single-cell data silos, and we can build these predictive models. And so the first thing that we did was to focus on immune function. So we stained the starting material with antibodies that detect the phenotype of cells, essentially cell surface antibodies. And then we can perturb these cells with drug and we can then repeat our staining to see how the phenotype changes. And then at the end of these repeated cycles to see continually how the phenotype changes, we can then do functional testing to understand which cells are responding, and to correlate these multiple time point phenotypes with cell function. And that's really important in different fields. For example, when we're looking at immunogenicity, we have to understand which cells are responding to a particular treatment. And the only way to understand that is by doing multiple measurements. In immunotherapy, we have to look at which cells have the desired function. And if we can predict that function just from phenotypic analysis, that would be incredibly powerful. And so that is where we're headed. We're trying to get these dynamic measurements of phenotype that can predict functional outcome. If we did this with regular conventional flow cytometry, with a series of different samples at different time points, we would connect the dots, but we would be greatly misled. So the study goal here would be to characterize T-cell activation. And what you see is that for PD1, CD25, and CD69, each of these markers goes up, and it looks like the entire population of cells is just going up. And that's how these markers are expressed in immune cells. Well, it turns out that when you do single cell analysis and you do kinetic analysis, time-lapse analysis using these optical barcodes, what happens is that you can see that actually the picture is much more complicated. So instead of CD69 going up and then being maintained, sure, there are some cells that do that, but there are other cells that go up transiently and then go back down. And there is still another set of cells that take a while for them to reach expression of CD69. So they're late expressors. And so now, using these dynamic measurements, we can distinguish 3 different profiles for the expression of CD69. It's no longer, is it positive or negative at a static time point? We can watch the movie of what happens with different cell types. Are they sustained, late, or transient in their expression? And we see for the other markers that we measure this same pattern, although the frequencies of these cells, the proportion of these different profiles is different. You do see that there are some cells that maintain, some cells that have late expression, and some cells that have transient expression of CD25. And similarly, for PD1, we see the same kind of thing, although the difference, the changes over time are not quite as large. So what then if we took a functional measurement at the end of these multiple time points and then tried to track phenotype to function, or P2F as the folks at Lay's call it? We can do that using this unique technology where we have basically PD1 expression at the first time point, at the second time point, at the third time point, PD1 or CD69 expression at each of the time points, and then we can lay out a table like this and then say, does this individual cell that has this characteristic of expression secrete or not? And we can build out an entire table to get all of this information for every single cell. And when we do that and we feed it into machine learning models, we can find that there are particular kinetic phenotypes, particular time points of cell surface protein expression that can predict function. And that's the case for TNF alpha. It can predict the function with a good AUC of 0.88, particularly good at the specificity. Interferon gamma and TNF co-expression can even be predicted with a high AUC 0.85 and much more sensitivity and specificity. So if we find a TNF alpha cell, what is the history of its expression over time? Well, it was likely to be CCR7 positive at 24 hours, likely to be PD1 positive at 48 hours. And when we go through this list, we can understand and build stories of how these cells change over time. So TNF alpha secreting cells are likely making CCR7, meaning they're central memory or naive cells at early time points in the course of stimulation. are likely also to be destined to become exhausted because they express PD-1 at late time points in the stimulation. And so you see how we can develop an interpretive story that's rich from this type of very deeply defined and well-characterized data. And I think that's the next horizon in flow cytometry. So today I've shown you how we are innovating at Talon Biomarkers at every turn. This is stuff no other CRO is doing, and I'm really proud of that because these are going to be the future of translational science, and we are bringing that future to our clients now. We can do the classical assays. We can do them in a way with our quality control tools, with our panel design, with our data analysis. We can do it in a robust, reproducible, incredible way. But we can also take that knowledge and extend it and allow our clients also to innovate in their space and to benefit from the great information content that we get from this innovation. So I want to thank the folks who work at Talon, Alex, our lab operations manager primarily, Ariel, who was our chief commercial officer, but she's moved on to become CEO of our sister company, TerraFlow, that I told you about. Aidan Kelleher, who is a scientist in the laboratory. Daria, who's a business development lead. I'd like to thank Dan, my co-founder at TerraFlow, Sheldon and Trevor, who are at the awesome Lays company that I think is really doing the most exciting work in flow cytometry right now. And it's really the future of flow cytometry. I think we're doing the most exciting work in the CRO space. And if you'd like to connect with me on LinkedIn, I've got a QR code here, and you can learn more about our work. We do CRO work, QC and panel building. We do data analysis. We can talk to you about TerraFlow, time-lapse flow cytometry consulting, and tech development, because we have a strong history in this space as well. So I'll leave it at that, and we'll take a few questions. We'll bring Christy back to basically moderate those questions. And we appreciate all the time that you took to listen to this, and are so excited for this opportunity to talk with you all.
Well, Dr. Chattopadhyay, thank you so much for this informative presentation. We have so many great questions that have already come in, but I want to remind our audience that if you haven't already done so, please do so now. Type in your questions into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for at the that time allows. So, Dr. Chattopadhyay, let's start right here with this question. So when adapting high-dimensional panels across instruments with differing optics and detector sensitivity, how do you validate that spectral resolution and marker performance? How do you validate them that they are preserved? And particularly when substituting dyes for compensating for unavailable conjugates?
So this is a really important question that ties into how we more broadly do our flow cytometry work. And so there's a lot of effort in the field to make instruments match each other, essentially, and to quantify the differences between instruments. That's a really important effort, and is really valuable. When the reality is, though, that science marches forward, and we may build a panel on one particular platform and then ultimately you need to use it on another platform. And in order to do that work reliably and carefully, you have to understand that there are a number of components to making that happen with care. The first is titration of the antibodies on each of the different platforms, because it may be that the sensitivity is so different that the amount of antibody that's needed to quantify the same population of cells is very different across these platforms. And so that's the first step to doing this reliably. The second step to doing this reliably is to understand the instrument configuration and the instrument performance to make sure that those are at least close across the different instruments. If you can't measure UV dyes because you don't have a UV laser, this is a pointless endeavor. The third area where we look at to make sure that we're thinking about reliability is to understand the biology. And that's really where Talon excels. That's really where we can help. We are fundamentally academics and scientists at heart. And so we go into the literature and we understand if the expression of the markers follows what people have seen previously. And we understand if it makes sense in the biology. And so you look at different types of samples, healthy individuals, stimulated samples, unstimulated samples, to help us understand whether the biology is true. And that's ultimately the best measure of this. So we can try to make instruments match exactly worldwide as much as we want. But if we're not considering the biology, then we're ultimately failing the science.
Thank you. Now, as you know, CLR transformations address the sum constraint, but how do you handle potential interpretability issues that arise from the log ratio space, especially when presenting findings to clinicians and cross-functional teams?
So, this is another important piece of how you interpret and explain results, and we do a lot of transformations of data in our work, and as scientists generally, data transformation is really important to help us understand what's happening in a disease mechanism or a process. And I find that it's easiest to go about explaining to people the basis for this transformation, just like I did in this talk, essentially, that there's a dependence between the different cell populations when you have just proportions, and that's all you're looking at. And so when we do these types of log ratio transformations, you can easily see that that dependence is lost. And actually, there is something appealing about the visualization because it's centered log ratio. If there's no change, essentially it kind of tracks around the center of the data, and because of that, it really emphasizes when there are changes, and that's very appealing to our translational science and clinical partners because it's almost like it's an easier way to visualize the data. Ultimately, all our visualizations are transformations of data, and as long as you can explain that reasonably well to the clinician, they are very accepting.
Excellent. Our next question. Thank you for this great presentation. Now, our high-dimensional data sets often suffer from redundancy and multicollinearity. How does your pipeline ensure robustness and generalizability when selecting predictive features for biomarker modeling?
So Terraflow is particularly good at this because the first thing we do is that we build out all the potential phenotypes that are present within a data set. And when you do that, when you're comparing those across patient groups, you're essentially introducing a false discovery problem because you have lots of different variables that you're testing against your outcome. And so we do a false discovery rate adjustment that allows us to consider that. And that's a common approach to deal with issues like essentially, doing a large amount of comparisons. The other thing we do is we do cross-fold validation. And so when we build our models that include all of the phenotypes that are significant after false discovery rate adjustment, we do a tenfold cross-validation where we build a model and then we take 90% of the samples from the data set, and we use that model to predict, to classify those. And then we do it on the remaining 10%, and we compare those, the specificity and the sensitivity. So we do this cross-validation. We do that 10 times and only report the results that are consistent across the tenfold cross-validation. And so that's how we deal essentially with this problem in two different ways. And I think it's probably one of the stronger ways to deal with this problem that's out there.
Thank you. We have time for just a few more questions. This next one is two parts. Did your team test how various unmixing algorithms influence the final flow cytometry results? And do these differences significantly impact the outcomes?
That's a great question. So to some extent, we have done this early on. We made sure that we did our work on instruments that were capable of both spectral cytometry and conventional cytometry, where one particular 4-chrome was assigned to a particular detector. And so we did standard compensation for all our studies. We did standard compensation, and then we did spectral unmixing from the instrument. And then we also did spectral unmixing from our data analysis program, FlowJo. And we found over time that we were getting essentially the same biological outcome. And so, at least in my hands, the unmixing algorithms didn't matter as much. They might, your mileage may vary. We have panels that are highly optimized. And so because of that, it may be less of an issue. But if you have a less optimized panel, this could potentially be a problem, and you might have to choose a particular unmixing algorithm. And then you go into the situation of trying to understand which is true and which is not true. And I think the way that you approach the answer to that is that you actually run your unmixing algorithm on the beads that are the single color compensation beads. So you concatenate those all into a single file, data file, and then you run the unmixing algorithm on those. And you should have a pure signal in every dimension, essentially, that is not showing up in the other dimensions. Then if you have one particular unmixing algorithm that doesn't have that purity on the beads, on the concatenated bead file, then you know that that's the wrong unmixing algorithm, and you don't have to rely on guesswork within your sample set. But this is really not a problem that we've seen, which is kind of cool. I think it makes it work easier.
Thank you. And Dr. Chattopadhyay, we have time for one last question.
Sure.
How do you ensure barcode signal integrity across sample prep, handling, and acquisition steps, particularly for long-term or multi-platform experiments?
So this is a question about the LACE technology, and they have extensively characterized these issues in the papers that are that were presented. You can look up the work, and you'll actually see those experiments in the papers. But what it boils down to really is that you need a material that is coded for biocompatibility, and you functionalize that with an antibody. So really, you're not doing anything different than antibody-based tagging, and we're doing that in a way that's very similar to quantum dots or other things that are essentially stuck onto antibody. And so there's not really an issue with integrity across sample prep, handling, and acquisition. And so that makes it, particularly reliable and robust to do that. Now, to be fair, there are some cells that you will lose in the process of handling, like centrifugation, but that's a cell loss problem that's common across all our platforms. It doesn't have to do with the optical barcode. And essentially what happens when we analyze the data is that you see for every barcode that's there, you check to see that you have data downstream, and you only report the data for which there is an optically barcoded cell that's present at all the time points.
Dr. Chattopadhyay, thank you so much for your time today and for your important research. Do you have any final comments for our audience?
No, happy. Thank you so much, Christy and LabRoot's team for setting this up. As you can imagine, we are passionate about the science that we do. We're really passionate about flow cytometry, about single cell technology, and we are eager to connect with you, whether you have a project now or later, or whether you just want to talk about your work. We are happy to talk with you and happy to connect with you. So look me up, shoot me an email, find me on LinkedIn, and I'll be excited to talk to you about your science. So thank you so much again, and have a great day, guys.
And thank you. And I do want to thank LabRoots and our sponsor, Talon Biomarkers, for underwriting today's educational webcast. Before we go, I also want to thank the audience for joining us and for their interesting questions. Questions we did not have time for today, and there were quite a few. And additionally, those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. That's all for now. Thank you so much again for joining us. And Dr. Chattopadhyay, thank you for joining us. Have a great day, everyone. Bye-bye.